very good morning to all the ladies and gentlemen. Today is Happy Teachers Day, and we begin by this shloka: Guru Brahma, Guru Vishnu, Guru Deva, Maheshwara, Guru Sakshat Param Brahma, Tasmai Shri Guruve. There are so many teachers today who have logged in as program directors, as chief guests, as discussants, and we just do Tasmai Shri Guruve Namah. Who can be a better than our program director today, Professor Dr. B Agarwal. Thank you, sir, for 4 decades of teaching, 4 decades of leading, 4 decades of inspiration, and 4 decades of creating bundles and bundles of hematologists today. It's an honor and privilege to get associated with Mumbai Hematology Group under your kind leadership. Over to you, sir.
Thank you, Dr. Manoj, for those kind words. I'll just share my screen. Good morning to one and all. Happy Teachers Day. This webinar, as usual, is brought to you by Mumbai Hematology Group, a very busy body as far as the education is concerned. It is supported by Intas, our regular partner, and managed by Mice Ideas as usual. I thank Mr. Dr. Manoj Kumar and his team from Intas for supporting our academic activities. Mr. Rajesh Kalpesh and their team from Mice Ideas for managing these shows flawlessly. Executive Committee of Mumbai Hematology Group. Our chief guest today, Professor Dr. Prantar Chakrabarti from Kolkata. Our guest speakers, Dr. Shaji from Velour and Dr. Regis Peffault from Paris. All our discussants are themselves eminent molecular hematologists, clinical hematologists, or hematopathologists from India. And you participants for sparing your Sunday morning. Our next weekend has got 2 fantastic academic activities. We have Dr. Verstovec, Professor, Department of Leukemia from MD Anderson, who will be a discussant for 11 cases to be presented by Indian faculty. So don't miss this on Saturday, 11th of September at 7 p.m. 11 cases with masterly remarks by the faculty, our guest. Next day, we have Dr. Flora from Italy, Milan, and she is going to cover a relatively rare subject of rare bleeding disorders. So genetic bleeding, but not hemophilia and not von Willebrand disease. Our today's guest speakers, as mentioned, was Dr. Shaji from Velour and Dr. Regis Peffault from Paris. It's my pleasure to introduce Dr. Shaji this morning. He obtained MSc Biotechnology 1993 Goa, and then PhD CMC Velour 2001, 2 decades ago. Currently, he is Professor of Molecular Biology at the Department of Hematology and Adjunct Scientist at Center for Stem Cell Research, CMC Velour. His career interests include developing the molecular methods to study inherited hematological diseases and research on genetics and molecular basis of red cell diseases and inherited bone marrow failure disorders using primary and induced pluripotent stem cells from the patients. He has over 60 peer-reviewed publications in international and national journals. Our discussants this morning include Colonel Dr. S. Venkatesan. He is MD Pathology from AFMC, DM Hematopathology from AIIMS New Delhi. He is Professor, Department of Pathology at Armed Force Medical College, Pune. Dr. Pooja Prasad, Assistant Professor, Department of Hematology, VMMC and Safdarjung Hospital, New Delhi. Dr. Aditi Shah, DNB Clinical Hematology, Department of Hematology at Narayana Hridayalaya, Bengaluru. Dr. Reema Miria Ibrahim from Department of Hematopathology, AIIMS, New Delhi. Dr. Dibay Prusty from Department of Clinical Hematology, SCB Medical College, Cuttack. Dr. Mona Vijayaran from Department of Clinical Hematology, AIMS, New Delhi. Dr. Akshay Mandaloy from Department of Hematopathology, TMC, Kolkata. Now we come to our regular postgraduate Sunday quiz. Today's quiz is related to DOACs, that is direct oral anticoagulants. We'll give you 10 statements about DOACs. One of the statement is wrong. You have to pick up the wrong statement. And the number of that wrong statement, that number 1, 2, 3, whatever the number is, that is to be emailed to mbagarwal1@gmail.com. And the winner will be fastest finger first. So the email that you have to use for forwarding your reply as 1, 2, 3, 4. You don't have to write the statement, is put at the top. Now I will read out the 10 statements, and one of them, which is wrong, you have to pick up and forward the number. 1. The terms DOACs and NOACs stand for the same compounds. 2. Dabigatran, rivaroxaban, apixaban, and edoxaban are all DOACs. 3. As against vitamin K antagonists, DOACs have improved therapeutic index. 4. DOACs are unsafe during pregnancy and breastfeeding. 5. DOACs do not require routine coagulation monitoring. 6. DOACs are approved for stroke prevention in non-valvular atrial fibrillation. 7. In long term, dabigatran and apixaban are given in BID schedule, while rivaroxaban and edoxaban are given as QD. 8. Rivaroxaban is usually given on empty stomach, that is fasting. 9. Idarucizumab, brand name Praxbind, is a humanized antibody fragment and is a novel antidote to dabigatran. And last, Adexanet alpha, brand name Andexa, is a recombinant inactive human factor 10A analog. It's a novel antidote to rivaroxaban, apixaban, and edoxaban. So, whichever was the false statement, number of that, email it to mbagarwal1@gmail.com. Now it's my pleasure to introduce our chief guest of the day, and we are honored by kind presence of our chief guest, my friend Dr. Prantar Chakrabarti from Kolkata. Well, thank you so much, Dr. Prantar Chakrabarti, for being honored to us. A brief introduction of Dr. Prantar Chakrabarti. He is consultant hematologist at Vivekanand Institute of Medical Sciences, Fortis Hospital, Kolkata, and Emory Hospital, Kolkata. He did his MD in internal medicine, then he did his DNB in internal medicine, and finally did his DM in clinical hematology from AIMS, New Delhi. He is a recipient of many national and international accolades, including Royal College of Physicians, International Bursary, Harold Benson Fellowship, and Marie Curie Action Fellowship. Besides, he has received specialized training in ethics of human subject research, biostatistics from Johns Hopkins University, Baltimore, and stem cell transplantation from King's College, London. He is a professional member of ASH, ISHBT, AI, and IMA. He was the scientific secretary for the 51st Annual Conference of Indian Society of Hematology and Transfusion Medicine, Kolkata, which was held 14 years ago. His areas of special interest include hematopoietic stem cell transplantation, thalassemia, and translational research. Dr. Prantar, I request you to inaugurate our webinar and give some words of wisdom to all of us. Thank you.
Thank you so much, sir. And lest people forget, my regards to one and all, and especially Professor Agarwal, who actually was the gatekeeper to my final degree in hematology. So when I think of those days, and I still cherish the way we have learned hematology, and I think on this Teachers' Day, it's a privilege for me to thank all the teachers. Sometimes we don't even realize that we are learning from others. And such has been the impact of the entire universe on our learning. I think hematologists per se keep their eyes and ears open more than other disciplines. And as I always joke with my students, that we are the best clinicians amongst pathologists and the best pathologists among clinicians, because that is how we have been molded. And the best teacher in the last 1 year, I think, has been this small virus. So if you look at this COVID pandemic also, people started thinking that ventilators will save lives. They were targeting the lung, they were targeting oxygenation. But until and unless the hematologists came into the scene and showed that there were clots everywhere, and we have to anticoagulate to save lives, lives were not being saved. So this is the impact of what hematology does. And so when people think, that these people, oh, what are they do? Some of my medicine students have told me, sir, it's a thankless job. You actually cannot see what goes beyond what we do daily. Even during this pandemic, let alone anticoagulation, when the vaccines were administered and you suddenly saw some side effects, who were the first to tell you that yes, something like TTP is occurring? And the first publication in AEJM was from a hematologist, Professor Andreas Greinescher, who, as sir mentioned, had come to HematCon in 14 years back. And that is how translational research is implemented in hematology. So what Professor Shaji will tell you or others will tell you going on in the lab, you would possibly be the first person to use it in clinical practice if you look at across disciplines. So it has been a pleasure. The goddess or god of hematology inspiring us to look at things, not only to follow protocols or go about in a routine way. You have to be innovative, be it in diagnostics, therapeutics, and even in big data analysis. So whatever you do in life, hematology will touch you and you can't run away from it. And I would like to thank Mumbai Hematology Group, especially Professor Agarwal and his team, for giving me this honor on a Teachers' Day to thank all the teachers in this world. Thank you very much.
Thank you, Professor Prantar, for those very kind words and very inspiring speech and putting hematology at the top of everything. I'm so grateful to you giving such credit to hematology today. Hardly anybody must have done this. Thank you. Thank you. And now we have the first guest lecture. And it's my pleasure to invite Dr. Shaji R Velayudhan from CMC Vellore, Professor of Molecular Biology and Hematology, to tell us about molecular diagnosis of inherited bone marrow failure diseases. Over to Dr. Shaji.
Is my screen visible?
Yes.
Okay. Thank you for this invitation. And I've been hearing about this Mumbai Hematology Group's meetings for a long time. And I'm really honored to have this opportunity to speak in one of these meetings conducted by the Mumbai Hematology Group. I slightly changed the title of the talk. I just added the word inherited because I'm going to focus on two important diseases in which we have done research and finally established molecular diagnosis. And one of them is Fanconi anemia. I will take you briefly through the research, then spend a lot of time on the diagnosis algorithm that we established in the lab finally. After 10 years, we have a fantastic molecular diagnosis pipeline established for Fanconi anemia. And then I will take you to a set of slides to show how the same strategy is extended for the diagnosis of diamond blackfan anemia. To start with Fanconi anemia, I'm sure I don't have to give a lot of introductory statements on Fanconi anemia. It's the most common inherited bone marrow failure disease. Although it's a rare disease, among the inherited bone marrow failure diseases, it's the most common one. In India, we see fairly large number of patients. At CMC Vellore, we see at least 50 new cases of Fanconi anemia every year. It's a big number. Therefore, we started research on Fanconi anemia 10 years back. And these are the developments that we did subsequently because the large number of patients that we have in our OPD clinic. And the carrier frequency is, you can see here, 1 in 200. This accumulated data from different populations. This does not say anything about the Indian population. What the message here is, it's a rare disease, but it's common in India. And the clinical features, the most common clinical feature that we observe in the patients with the Fanconi anemia is progressive bone marrow failure, which is observed in more than 90% of the patients. And these patients have congenital malformations. This list of malformations that you can see in these patients, and that is typically seen in 75% of patients. Still, we have to remember that in 25% of patients, these congenital malformations may not be seen. So there, with patient comes with bone marrow failure without congenital malformations, a differential diagnosis is required to differentiate aplastic idiopathic aplastic anemia and Fanconi anemia. And one important factor that is in Fanconi anemia is that there's an increased risk of malignancies, including MDS and AML, that goes up to 30%. And that may come later in life, and we can't wait for this MDS or AML to come for the differential diagnosis. And the most predominant phenotype that is seen in this patient is the sensitivity to DNA cross-linking agents. So if any drug that is given, which is a DNA cross-linking agent, then you can see that the patient is reacting to it. And that is an indication that patient has Fanconi anemia. And so if there is an aplastic anemia patient or patient with leukemia or MDS, when it's treated with any DNA cross-linking agent drug, then the patient has got adverse effects, then that immediately tests have to be done for Fanconi anemia. I'll go more into details why laboratory diagnosis is required in the subsequent slides. Before that, just to tell you that there's a very complex DNA repair pathway that is involved in Fanconi anemia. There's more than 22 genes, and that says that the molecular diagnosis is very difficult, and you have to screen all the 22 genes finally to give a molecular diagnosis. And they have functions there. There is an upstream complex of proteins, and they are responsible for ubiquitinating FANC-I and FANC-D2. It's an important step, and this is used for the molecular diagnosis. I'll come to that later. And this ubiquitination of FANC-I and FANC-D2 is responsible for the DNA repair to happen. This happens in the normal cells properly. If anything happens to any one of the genes in the upstream complex, or the FANC-I or FANC-D2, which does the recruitment of proteins in the DNA for DNA repair to happen, in the downstream proteins, so mutation happens any one of them, then what happens is the patient will have Fanconi anemia. And there's the sequence of steps that happens at the molecular level at the DNA and the chromatin is very well studied. There's a lot of studies still going on to understand the fine mechanisms. So there is upstream proteins, there is FANC-I and FANC-D2 ubiquitination, then downstream proteins. So there are three components in the FA pathway. So in addition to this, the FA pathway, just to tell you this strong biology here, there is this FA pathway, the genes, the 22 genes, in addition to be in the DNA repair, and they also have additional functions. And this is responsible for the phenotype changes that we see, variety of phenotypes that we see in the patients with the Fanconi anemia. Because the patients with the FANC-A mutation, patient with the FANC-C mutation, patient with Fancane mutation, they may have additional phenotypes in addition to the bone marrow failure. So this is an important factor when you do the molecular diagnosis of Fanconi anemia, that there can be mosaicism in the patients, and this 25% of FA patients, they show somatic reversion in hematopoietic cells. So the hematopoietic cells alone get corrected, and this leads to mosaicism. And okay, and it's caused by spontaneous reverse mutations that restore functional FA alleles. Therefore, when you go and do the molecular diagnosis in the peripheral blood, you have to be very careful, because those hematopoietic cells may be getting corrected in, and that occurs in 25% of the patients, as I said. And mosaicism can give a selective advantage, and the corrected hematopoietic stem cells can improve the clinical outcome, but that happens only in the hematopoietic system. And that's called natural gene therapy. Again, it happens only in the hematopoietic cells. And a small cells, at least one cell, if it gets corrected, because of the survival advantage of the corrected cell, and that cell can overpopulate and can make a large pool of normal HSCs in the patients. And this is very, very important. We look at very carefully whether the patients have any signs of clinical improvement in the hematopoietic cells before confirming the molecular diagnosis. And subsequently, it's very important news is that this mosaicism factor and the science that learned from mosaicism has been used for gene therapy now. Now, why is it important for accurate the lab diagnosis of Fanconi anemia? Some of those factors which I said earlier, and I'm restating them here. 25% FA patients do not have FA characteristic physical abnormalities. So therefore, it's very difficult to differentiate FA from AA, aplastic anemia, when we do the diagnosis, especially for young children. Then the differential diagnosis between AA and FA is important for deciding the transplantation or for ATG treatment that is done for the AA patients. Then the children with leukemia, MDS, or solid tumors may have underlying FA, and they may not tolerate the unusual dose of chemotherapy drugs. I mean, the usual dose of chemotherapy drugs that's given to leukemia and MDS. So this has to be looked at very carefully. If there is history of Fanconi anemia, and then that has to be checked, and it is important to rule out the Fanconi anemia in the patients. And 15% of the FA patients, as I said in the previous slide, they have somatic mosaicism with normal hematopoietic cells. So therefore, in the hematopoietic cells, it may look, the counts may be improving because of the somatic mosaicism that is setting in within the cells, within the hematopoietic cells. Therefore, it is important to rule out the Fanconi anemia patients. So then, when there is a patient with FA, it is important to do prenatal diagnosis and genetic counseling, and that has to be done in the screening by screening the siblings. These are three important tests that are used for the laboratory diagnosis of FA, and one of them is chromosome breakages analysis. And that is, I will go through the details of the tests when I talk about what we did, and take the peripheral blood lymphocytes and add mitomycin C and look for chromatin, chromosome breaks, and document them using variety of formula, and then you can find out whether the patient has FA or not. And then cell cycle analysis is a useful method, and several groups have used looking by cell cycle analysis, see whether a large number of cells are, whether they have G2M phase arrest or not. And we mostly use the FANG-D2 ubiquitination as a method. I'll tell you the details why we use it. But just want to state here that there's a third method which can be used for the diagnosis of Fanconi anemia. As I said earlier, FANC-I and FANC-D2 ubiquitination is an important step. Any mutations that happen in the upstream pathway genes that will affect the ubiquitination of FANC-D2. And ubiquitin band will migrate in the Western blot as an additional band, and that will be lacking in the Fanconi anemia cells. And that is very easy to do the diagnosis without doing a lot of interpretations. You do not have experts looking at the chromosome breaks and doing calculations, etc. And I've given the advantages and disadvantages of this method. Chromosome breakage analysis is sensitive. However, needs an expert, and it has to be looked at very carefully, especially when there are borderline values. And there are a lot of communications you have to do with the clinicians to see, hey, I'm seeing only borderline value. What is your clinical phenotype? And so a lot of discussion and judgment call has to be made finally. And flow cytometry analysis for the G2M phase arrest. As I said, it's for looking for cell cycle analysis. It is easy to interpret and relatively simple assay. Having said this, although the textbook says like this, we took a long time to establish this method. And there are a variety of methods available, and it was not easy for us. And I'll show you some data that we finally have for the G2 arrest documentation in the FA patients. However, if the patients have for leukemia, the G2M arrest will not look, is not suitable for the diagnosis of FA. And if the blood cells are used, again, it is not suitable. And because there will be restoration, phenotypic restoration, and there will be a correction, and the normal cells will be present. And this is the FANC-D2 ubiquitination test. Okay, I'm not sure the screen is visible fully. Can you see the full screen?
Yes,
we can see.
Okay, and the FANC-D2 ubiquitination, what we do we did for large number of patients, and it's easy to interpret, and it's very specific, and it is possible to do the diagnosis in 95% of cases if we include skin fibroblasts also. And I will take you through the algorithm in the subsequent slides. But as I said earlier, it is not diagnostic in 5 to 8% of the cases because the downstream proteins are not, they do not affect FANC-D2 ubiquitination. Fortunately, the downstream genes and the mutations are rare in FA disease. And the two types of cells are required in the lab, and one of them is the EBV immortalized B cells, and we routinely make them, and we have standardized this protocol very well. It's all, when somebody wants to establish this, I don't want to tell that this is so difficult now, but I just want to tell you that how systematically we did it to finally make a diagnosis algorithm. And this is EBV immortalized cells, and whenever possible, we got skin fibroblasts, and skin fibroblasts usually collected when the patients go for bone marrow biopsy, and anyway, the patient is in anesthesia, and that time, after informed consent, we take skin fibroblasts from the region. And when we did this analysis, and several times we had to confirm that the cells are Fanconi cells, we did this conventional complementation methods, and in here we cloned the Fanconi anemia genes. Here you can see here FANC-A, FANC-G, FANC-C, FANC-D2, then subsequently FANC-I, L, and several of these lentiviral vectors we made, and then we made these lentiviruses, and then what we did was we transduced them in the lymphoblastoid cell lines or fibroblasts, then look for phenotype correction within these cells. And for phenotype correction, we looked at chromosome breakage analysis to see whether chromosome breakages are reducing when mitomycin is added. Then do a cell cycle analysis, or FANC-D2 Western blot has been always our favorite method. So in brief, these lentiviral vectors which code for the normal copy of the gene, the viruses are made with these lentiviral vectors. Then we transduce the lymphoblastoid cell lines or fibroblasts from the patient and see whether there is any phenotype correction. So that gives an idea that when the cells were transduced with the FANC-A gene, if the phenotype correction happens, FANG-A gene was defective in those cells. Similarly for FANG-G and FANG-C. And this is the methodology how we developed. And after clinical evaluation, I'm going to take you through all the steps that I have already said. And after the Fanconi anemia features documented, doing chromosome breakage analysis, and look at whether the patient has low scores or high scores or borderline scores, then do FANG-D2 ubiquitination Western blot. I told in the previous slides that has been our favorite method, and subsequently we take the cells for exome sequencing. And in the peripheral blood, if it is negative, we take fibroblast and see whether the patient is negative or positive. If it is negative, we'll discuss with the clinician to see whether we should proceed further for doing exome sequencing or not. Otherwise, it's very expensive. Data analysis going to happen in the downstream steps. So we have to we discuss with the clinicians to see whether we should proceed further or not. Then novel mutations, when they are identified, we do the complementation, which I described in the previous. This is for research, and now we do not do lentiviral complementation. We have come out of those stages of confirming and whether this mutation is pathogenic or benign or not, and that is complementation was required for establishing the complete pipeline. But now it is not required anymore. And after that, we look for the complemented cells. We'll see whether there is phenotype correction or not. And here, I just want to mention that exome sequencing cannot robustly detect point mutations. Therefore, we included MLPA. It's a method for detecting short deletions within the genes. So just want to say that finally, what did we get after doing the molecular diagnosis? Before telling the current pipeline that we have in our lab. And as I said earlier, we did a large number of patients. We did chromosome breakage analysis, interpreted, look for tri-radius, and look for the breaks that occur. Then do Western blot. See, you can see here the control T cells, 2 bands are there because there is a normal ubiquitination of FANG-D2. Ubiquitin band will make the second upper band. And the patient fibroblasts and T cells, we can see here there's only 1 band. It's very easy to interpret. And then looking at the chromosome breakage scores that we have from here and how it is distributed among patients with Fanconi anemia and aplastic anemia and control. You can see here there's significant overlap here, and this large number of FA patients have normal CBS scores. Then categorizing them based on the CBS scores and the Western blot and see whether these patients have upstream mutations or downstream mutations before doing the NGS. And this is cell cycle method which we recently established after a lot of struggle. Now looking back, we don't know why we had to struggle so much. And you can see here is a normal control cells. After adding mitomycin at different concentration, there is no difference in the G2M percentage, the cells at G2M stage. And here, the 2 patients LCS, when mitomycin is added, you can see here that G2M percentage is steadily increasing. And the 2 LCS that we had, and these LCS were made from patients with Fanconi anemia. So this method now looks like it's working because it's in a range of concentrations of mitomycin C, and it is clearly showing the phenotype. Now we think that this is a very good method that within 4 days we should be able to do a cell cycle analysis and tell the patient that tell the clinician that the patient has FA or not. And we have it included this in our the algorithm, and we're going to do for more number of patients. And if this is going to work with the peripheral blood in 4 days, we will be really happy, and we'll be able to do this lab diagnosis for a more number of patients. And this will be the concentration of mitomycin C that we are going to use. So essentially how it works is take the blood cells, add mitomycin C of one particular concentration, and you have the normal control and see whether there's increased G2M cells at G2 phase or not. And now I'm going back to the molecular diagnosis of Fanconi anemia, where we had around 150 patient samples we have done by NGS. And this is the pipeline, and which a lot of discussions we had to do with the genome bioinformatics team, and who has been very, very helpful in sorting out many of the doubts that we had about the pathogenic mutations and variant with unknown significance, benign mutations. Finally, some of them became pathogenic. And we learned a lot from this discussion. Now we are very confident that we'll be able to tell if the patient has got pathogenic mutations or not. And as I said earlier, if there's a case in which we can't make out whether the patient has got pathogenic or benign, which drop down to 1 or 2%, now we do a complementation and confirm. But that is, we find that that's very rare. And I'm not going through details of the bioinformatics analysis here. Something very important steps here, as the machine gives a FASTQ data, and that goes to different steps here. And finally, the point mutations are classified, and then when the point mutations are filtered, to look for the population frequency and see whether these mutations affect splicing nonsense, whether they cause stop loss or stop gain and frame shift mutations, and whether they are not synonymous damaging. Based on all this, we call them as mutations which are associated with Fanconi anemia in the patient. And in this exercise, something that came up and we realized very important was it is possible to do copy number variations also. And this copy number variations are deletions that occur in 2 or 3 adjacent exons, maybe in heterozygous state or homozygous state. With a lot of research and analysis with Medgenome, we had a lot of data. We knew that those patients had MLPA, the patient had deletions by MLPA, and we used them as positive controls. And subsequently, in 100% of the patients with deletions, we could do molecular diagnosis by bioinformatics analysis also. And so far, which was done for around 32 cases, bioinformatics analysis helped in identifying the deletions. And this is the overall distribution of the mutations. Mutations, the type of genes that we have found the mutations. FANC-A has the maximum number of mutations, followed by FANC-L, FANC-G. And please look at this numbers carefully, because in our algorithm, we had to prioritize the genes. So FANC-A is the most common. And in India, FANC-L seems to be the second most mutated gene in Fanconi anemia. It is not FANC-C that is reported in the other population. So we had to do all this research finally for making the diagnosis algorithm, then FANC-G. And this constitutes the bulk of the FA patients here. And this, the heterozygous state is 5%. We did not see any patient without any mutations, but we found 5% of patients heterozygous state, and others are mostly homozygous because the consanguinity in our population, and we get large number of patients from South India, and with compound heterozygous state relatively less. And what is to note here is that we do not have a single patient without any mutations at all. And the distribution of the mutations, splicing, missense, frame shift, deletion, and nonsense mutations. And stop loss was very, very low. And MLPA is very, very useful. And we found that more than 20%, 18.8% of FA patients have deletions in one gene, which is FANC-A. And therefore, we thought, okay, we should have MLPA as a method. This is just to tell you the principle. Please do not look at these numbers here. I have the actual numbers in the subsequent slides. In MLPA works a complex method, but it's easy to do because we can get this reagents as a kit from MRC Holland, and they have the they have a kit for looking for deletions in the FANK genes. And MLPA, we realize it's something very, very important that we should do in our lab for FANK gene. And if you have any questions on the MLPA principle, I can come back to it. And this is to show you that how we compared the bioinformatics analysis that is showing deletions in this region. You can see a drop in the normalized reads from the bioinformatics, showing that there's a probability for a deletion. And MLPA showed the same region has got deletion in the heterozygous state. And CNV analysis by bioinformatics also showed there's reduced signal from that region. And this one, another region, there's a complete loss of 2 or 3 adjacent exons by bioinformatics analysis. And MLPA also showed that deletion of successive exons in FANK gene. And in short, if carefully done, bioinformatics analysis can identify copy number variations. Here I'm talking about deletions. And subsequently, if required, you can do an MLPA and confirm. This, all the patients that we did for the study that we performed, there's an MLPA showing deletions in large number of patients. And for diagnosis point of view, it is easy because these deletions we found only in the FANK genes. And one important information, as I said in the one of the previous slides here, is that the mutations in FANK-L gene, and this happened to be the second most gene with mutations here, which is not reported in other populations at all. And it became this clinical phenotypes of those patients, which are not different from patients with other patients with mutations in other genes. We completely looked and characterized and made sure this mutation is affecting the protein expression, and we have included them as a priority gene in our mutation analysis. We made several complementation vectors. I'm not going to the details of those vectors because it was important for us to have those for making our research successful in the development of a pipeline for FA molecular diagnosis. Because the FANK-A and FANK-G are the FANK-A is the topmost, FANK-L is 1 point mutation which can be detected by sample sequencing, and FANK-G has got unique mutations every family. Whenever FANK-G is mutated, we thought what we would do a long PCR followed by NGS for FANK-A and FANK-G instead of doing whole exome sequencing. We took 26 samples in which we found mutations in FANK-A and FANK-G, and we passed them through doing this PCR, then gave them for NGS. It is very, very cheap and it is not expensive, and we developed this method for detecting the mutations there. This is to give you a complete idea. I can go back to any of those slides if you have any questions. If there's an FA patient, because FANK-L mutations constitute 20% of the patients, and MLPA for FANK-A is also around 20%. What we thought was, as soon as the DNA extracted, do a sample sequencing for FANK-L and do MLPA for FANK-A. Within 3 days, we have data from around 35 to 40% of patients, and subsequently do an LA NGS for FANK-A and FANK-G, which is, it costs only 1,500 rupees for doing the sequencing, and it's only 56, it raises by another 56%. So that actually will boost up to around 80 to 85% of patients will be able to do the molecular diagnosis in 10 days. And we go to exome sequencing only if the mutations were not identified by these methods. And this entire diagnosis is become faster and cheaper. It's become affordable for the patients with FA. And we are currently validating this pipeline in more number of patients, and so far it's going on well. And just want to briefly touch upon diamond blackfan anemia. I spent time on describing the molecular strategies and research we did for finally establishing the molecular diagnosis for Fanconi anemia. Now, when we moved on to diamond-blackfan anemia, it became easy. There's a lot of communications with clinicians and pathologists and the molecular biologists reduced, and we know, okay, this is diamond-blackfan anemia, and the standard um, evaluation forms filled, and pathologists look at the bone marrow and see whether there's scarcity in red cell precursors. Then we move on to do the molecular diagnosis. And it's diamond-blackfan anemia, just to give a brief introductions, it's moderate to severe macrocytic anemia. Reticulocytopenia is one of the features, and normal bone marrow cellularity, but there will be a scarcity of red cell precursors. That is an indication that severe anemia with scarcity for red cell precursors is an indication for diamond-blackfan anemia. Correct me if I'm wrong, because I'm not a pathologist, I'm a molecular biologist. I learned all these things in the discussions with pathologists and reading the textbooks. And it's an important factor which you have to look at, whether the disease, this anemia, is congenital or it came later. And DBA usually presents in early age. And there's a positive family history is also supportive. And then all other minor evaluations can be done looking at elevated erythrocyte adenosine deaminase activity. However, this becomes difficult because most of the patients come to us, they're transfused, and congenital anomalies are seen in a small number of patients. They can be documented, and elevated fetal hemoglobin seen in more than 80% of patients. However, this is not possible if the patient is transfused, and no evidence of any other inherited bone marrow failures. Then we can we can consider that for molecular diagnosis for DBA, and no evidence of parvovirus infection which causes PRCA without underlying DBA. And DBA genes, we know that it's autosomal dominant. The mutation is seen in the heterozygous stage, and that is enough to cause the disease in the erythroid lineage. And that's called haploinsufficiency. And the mutations occur in the ribosome biogenesis proteins, and they are they are required for the translation of essential proteins within the erythroid cells. This is a recent paper which came in 2018 looking at the number of genes which are defective, and you can see here most of the mutations occur in RPS19. And there's a distribution saying that there are mutations can occur in the genes which are outside the ribosome biogenesis pathway. This includes SERC1, GAT1. These are rare, but they're also seen. So this is the data from our lab. So it was easy for us, and it's got more than 80% of patients with a suspected diamond-blackfan anemia. And we took them for NGS, and we found disease-associated mutations in 34%. Slightly less than 50% of cases. And we are taking them for whole exome sequencing. This was done in the focused exome. We are going to whole exome sequencing to see whether we can find mutations in all the patients. Here also, this is the y-axis represents the percentage. RPS19 constituted 50 to 60% of the patients. They're all in the heterozygous state. And the rare genes we found in 1 or 2 cases with mutations in GATA1 and SERC1. This is an important test that we realized this is not possible for routine diagnosis. However, as it's important to tell the clinicians as DBA or non-DBA based on the disease responsiveness or drug responsiveness this patient's group, to decide the patient has to go for transplantation or continue on drugs. When we find a case with suspected DBA, and we are not sure that it is associated with the mutation associated DB or not. Rarely we do an erythroid culture. And when we take the CD34 cells and culture with medium for erythropoiesis, there will be a differentiation arrest that can occur in the patients with DBA. And we have used it and for a patient who was going for transplantation, and we found that this test is useful. However, we know that it is not possible for routine diagnosis, but there was a case and we had to give confirmation. This was a research test that we developed in the lab through a project. And in the culture, they show a reduced proliferation. That is also an indication to say that the patient has got erythropoiesis maturation defect in the culture, and it really helped the patient. And DBA also, in addition to AGS, which I said here, we found that large number of patients can have mutations, deletions, which can be easily diagnosed with MLPA. This again, I really like this method. Sometimes it's a lifesaver, and we could give important information to the patients here. And the two families in which we have done the diagnosis here by erythroid culture that says that the patient has got erythropoiesis differentiation defect. Okay, and I will stop here, acknowledging the people who worked with me in developing here, and a lot of people worked with me 10 years back have left. And currently, Anurag Datt Chaudhary and Panindra, they're working with me in DBA and putting together the diagnosis algorithm. And a lot of support from Biju, Fauzia, and Uday Kulkarni from the clinical side in shaping up all these projects. And in all these occasions, I make it a point to thank Dr. Mamuchandi for inspiring me to work in bone marrow failure disease and making me recognize that this is an important area for research and development of diagnosis. Thank you.
Thank you, Professor Shaji. That was fantastic. So much of research, so much of work, so much of innovation. Thank you so much for putting it across to all of us. So there are faculty, almost 7 of them here. If anyone would like to ask questions, just raise your hand. So we have Dr. Venkatesan. Please go ahead.
Yeah, thank you, sir. And Dr. Shaji, that was an excellent overview, and it was great showcasing your work which is happening there at your center. So my question was, you have cell cycle analysis which can give you your diagnosis in 4 days. You have NGS and you have a whole exome sequencing nowadays. So in this era of these newer diagnostic modalities, what is the role of stress cytogenetics? Do we still need it? What's your take on this?
Yeah, most of the time, I don't want to go into a debate on that because if you have 100 aplastic anemia patients, if you put all of them through FA molecular diagnosis, lab will really get stressed. And that is not correct. So you'll be able to do a large number of cases. The stress test will filter them out.
Okay.
And you take them where there's a borderline cases, chromosome breaks there, and no chromosome breaks are seen, but clinicians think that there can be a possibility of Fanconi anemia based on the physical abnormalities. It's a family history, and those factors can be considered. Then we take them to Fanconi anemia. But however, I strongly suggest that chromosome breakage analysis has to be done. That should be in the front line.
Okay. Okay. So one more question for you.
Thank you.
So
can we take other questions first? Okay, Dr. Mona. Unmute yourself, Mona.
Sir, I'm unmuted. Am I audible now?
Yeah.
Yeah.
Yes, sir. So excellent presentation, sir. So my question is, why does a clinician, why is it important for a clinician to know the copy number variant, sir? Why should we?
The copy number variation is for us to give a molecular diagnosis to see whether there's a mutation or not. A clinician, it doesn't matter whether the mutation is a nonsense mutation or a frameshift mutation or a deletion. Okay, for the lab has to establish this diagnosis, and we have to know, not the clinician. It doesn't matter for clinicians.
Sir, actually, there are some pathogenic copy number variants which are more known to have more non-hematological manifestations, carcinomas. So some articles were stating that, sir.
It's not audible, Mona. Can you just?
Yes, sir. So actually, I've read some articles which were saying that the pathogenic copy number variants have more of extra have non-hematological manifestations in these Fanconi patients, sir. So is that, did you find any correlation?
See, in Fanconi anemia, any mutations which make null alleles, okay, whether it is a copy number variations because of deletions or insertions, or nonsense mutations or frame shift mutations, they all have the same effect in Fanconi anemia. And other situations, I agree, copy number variations have different roles, but in Fanconi anemia and in DBA, caused by null alleles, null mutations, I mean, this is not relevant.
Thank you, Dr. Mona. We have a couple of more minutes left with us.
Thank you, sir.
Thank you. Dr. Prusti, your question. Unmute yourself.
Thank you, sir. My question is about the mosaicism seen in Fanconi anemia. Sometimes these decoxybutan or mitomycin studies that comes negative in this Fanconi anemia patient, even the Fanconi anemia allele is present, but the mitomycin studies becomes negative. So how could explain this kind of mosaicism seen in Fanconi anemia?
Yeah, it's difficult. It's a challenge. That's why we say the mosaicism. You can do a chromosome breakage analysis in the fibroblast, or you can do a Western blot and check it. Okay.
Thank you. Rima.
Good afternoon, sir. So my question was, if children or if a young adult comes with MDS or leukemia, should we routinely go in for molecular testing for Fanconi genes, sir?
That is difficult. Probably, you realize that we cannot do that for all of them. If there is an indication when any drug is given, DNA cross-linking agent during the treatment, then you have to be cautious and also do a family history and check it whether there's any indication for Fanconi anemia-like disease. It is better to do it. That is what our department Biju suggests. And he says that it has to be looked at carefully. We cannot do for all MDS and AML patients.
Thank you.
Thank you, sir.
Aditi, unmute yourself.
Thank you, sir. So usually in a young adult with a severe aplastic anemia, we rule out PNH and inherited bone marrow failure syndrome. Suppose if they have a small PNH clone, should we still rule out an inherited bone marrow failure syndrome in the presence of an acquired mutation?
I will pass that question to Dr. Agarwal. I don't understand everything in hematology because I'm a molecular biologist. So how do you do a differential diagnosis and come if Dr. Agarwal has to send a sample to me? Okay, and I will ask him.
In the interest of time, I'll come back to you at the end of this webinar. Okay, let's finish with Dr. Shaji. Dr. Akshay.
Thank you so much, sir, for providing me this wonderful opportunity. And thank you, Shaji, sir, for a lucid and informative presentation. The only doubt I am having now is, do we have any false positive cases in chromosomal breakage analysis via mitomycin C, the other rare chromosomal breakage syndrome? Can they show positive results?
Yeah, there are. Okay, other disease, that's one of the problem with the chromosome breakage scores when we use that for molecular diagnosis, but fortunately or unfortunately, those diseases are much rarer than Fanconi anemia. Okay.
So in these cases, should we go for DEB rather than mitomycin C?
In our experience, mitomycin C is sufficient, and typically what we look at the triradials which are formed. Okay. In other disease chromosome breaks, we may not see so many triradials. Okay, mitomycin C is enough. DEB, getting it in India is very difficult. It's prohibited.
Thank you so much, Professor Shaji. Unfortunately, that's all what we could accommodate in the QA session. Okay. And if there are guys who have more questions, they will write to you an email, and you know that you will.
Okay. Thank you so much, sir. Thank you.
Thank you for sparing your morning today. Thank you. So we will now, I'll share my screen and tell you the answer of the quiz, and then we'll go for the second guest lecture. Right, so giving you the answer of the Sunday quiz. As you remember, this quiz was related to DOACs, and you were given 10 statements. And I'm not going to read these 10 statements again. 9 of these were correct. And the statement that was wrong was rivaroxaban is given on empty stomach. That was the only wrong statement. Rivaroxaban is best given with food or after food. So 8 was the answer expected from you to pick up the wrong statement. We had 12 correct answers and 11 of them are here. In the order of answering, Dr. Ramesh B, Dr. Dhanuksha Desanayaka, Dr. Sarthak, Dr. Virtisha, Dr. Krishnaratnam Kanand, Dr. Giri Punja, Dr. Thanuja Desanayake, Dr. Ketan Nathani, Dr. Andri Kurniavan, Dr. Radhika Sethi, and Dr. Mayat Marier Shui. So thanks to all of you. You did a fantastic job. And I mean, it's in the order. So some of you just missed by a whisker. And now to tell you the fastest finger fast in the correct answer. And that's Dr. Mona Vijayaran. She's a senior registrar doing DM in clinical hematology at AIIMS, New Delhi. And in fact, she's our faculty today over here. So congratulations, Dr. Mona. And we will get back to you with your prize. Thank you for participating and thank you. It must be wonderful. You have to be phenomenal to be fastest finger fast and correct. So congratulations. And now it's my pleasure to introduce our second guest speaker this morning, Dr. Regis Peffault D. latour. He was our guest speaker a couple of months ago. He had also spoken to us last year, and I'm extremely grateful to him for sparing his time and making us more knowledgeable in this part of the world with all the research and work that he has done lifelong. To introduce him formally, he is Professor of Hematology at the Hematology and Bone Marrow Transplant Department, St. Louis Hospital, Paris. He received his MD degree from the same university in 2003 and a PhD in Immunology from Pasteur Institute, Paris, 2006. After training in hematology in Paris, he completed his postdoctoral fellowship at the hematology branch of the NIH, Bethesda, from 2008 to 2010. He obtained his tenured professorship position in 2014. He's responsible for the French Reference Center for Aplastic Anemia and PNH, as well as the French Network for Immunohematological Rare Diseases. He's chair of the Severe Aplastic Anemia Working Party of the European Society for Blood and Marrow Transplantation and chair of the Bone Marrow Failure and Hematopoietic Disorders Subnetwork of the European Reference Network in Rare Hematological Diseases. His research interests include bone marrow transplantation, bone marrow failure, and paroxysmal nocturnal hemoglobinuria. He's authored over 300 papers cited in PubMed. He's a regular peer reviewer for journals like Blood, Hematologica, British Journal of Hematology, Biology of Blood and Marrow Transplantation, and Bone Marrow Transplantation. With great pleasure, I invite him to deliver his lecture today on paroxysmal nocturnal hemoglobinuria, an update on new treatments. Thank you, and over to Professor.
Thank you very much, Dr. Agarwal, for this very nice introduction. I guess you see my screen now.
Yes.
So it's my pleasure to be with you today, and really congratulations for all those fantastic meetings that you're organizing. I mean, it's really terrific. And what I would like to speak about, and what you asked me to speak about this morning, was about new treatment in PNH. And I would say that it's a field that is at a level of a dynamic completely crazy. I mean, this is, as you will see, this is a revolution for us to have access to so many drugs. But I guess that it's quite difficult from outside. When you're in the field, of course, you have access to those drugs. So that's and you're participating to trials, because we have, for example, 200 patients online with PNH. So of course, we're participating to all of this. But I think that from outside, it's quite difficult to follow because all those molecules are coming first row, and we don't really know where we are and where they are. So what I would like to do in the following 45 minutes, it's to give you a flavor of the most important data that we have in hands at the moment, or that will be published very soon, and also to clarify your mind on what drug is on development and what is the stage of each drug, because I guess it's really difficult to make a own opinion clearly, since, as you know, pharma are communicating loudly and they are not always the best ones to help us to make a kind of hierarchy on science and what we know about drugs, scientifically speaking. So basically, those are my disclosures and the company I'm working with. And as you can see, there is many PNH company, Novartis, Roche, and others, because of course, those drugs are coming to our institution and we're participating to trials. So the first part, quick part of my talk would be to remind to you, of course, the revolution that has been done with eculizumab in our patients. Our PNH patients were dying because of the disease at the very beginning of 2005. Patients were, as you know, presenting PNH with aplastic anemia, hemolysis, and thrombosis. And while we did a lot of progress in the last 25 years dealing with aplastic anemia, introducing immunosuppression, 91 Freecoffin paper, New England Journal, ATG plus cyclosporin, and transplantation that did a lot of progress in the last 20 years, especially for the introduction of the high allelic HLA typing. On the other side, the hemolysis patients were still suffering from thrombosis with devastating complications. So eculizumab for those patients where hemolysis was first row, and that's those patients that I'm going to speak about, were clearly with an unmet need, and we should recognize, and it's really something that we live on a daily basis, the drastic modifications of our patients under eculizumab. As you can see here, this is the two trials that has been published in the New England by Pete Hillman in 2004 and 2006 that were showing that these drugs in a randomized way was able to make patients less transfused, and more importantly, TE is thrombosis event rate down right. As you can see, under treatment, patients were protected under thrombosis. And that was really something really helpful. And then, of course, in the last 15 years, we saw that the patients were coming back to a normal life. And that was also working in real life because, as you know, most of the time, I mean, sometimes should be more precise, we are running randomized clinical trials. We're selecting the patients, and we're in a way not biased because this is a clinical monitored control trials, but we are choosing the patients, selecting the patients. And as you can see here, those are real life data that was published 10 years ago by the UK group. And that was basically showing that in real life, eculizumab was still doing great. And on the left side, you see that the overall survival of patients treated by eculizumab because of hemolytic PNH was comparable with age and sex match normal populations as a control. So in other words, eculizumab was able to make patients live the same way, I would say the same time, because the way is a bit different, speaking about quality of life, but at least the overall survival was comparable between patients with or without PNH under eculizumab. And again, because we were reducing the median time, the median transfusion rates, and because we were protecting from thrombosis. These things we also confirmed on a French study that was published 5 years later. And basically what we did show is exactly the same, is that eculizumab was significantly improving the overall survival of patients with hemolytic PNH. On the left side, you have 3 overall survival curve. It's all patients with hemolytic PNH. The black ones are those ones who has been treated before eculizumab was available. And the gray one was the one that was published when was the patients receiving eculizumab. And what we did is that we just compared those ones and matched them with patients with the same severity, with the same hemolysis rates, and we'll see what's going on. And on the other side, why there was 2 cohorts in black as historical controls, it was because in 85, we introduced the intensive care in the hepatological field, and then we were able to treat our Budd-Chiari syndromes with TIPS, as you know, and that was improving overall survival, but that was not something that was fantastic. And as you can see, before the TIPS techniques was available, after TIPS technique was available, and finally, when eculizumab was available, it saved many more lives. So we had a great treatment, and we should recognize this. There is no problem. Eculizumab was a revolution in the field. From 2005, it saved many lives. I showed you the effect on transfusions, on thrombosis occurrence, but it's also the case on pregnancy. We published this in the New England Journal, showing that women with hemolytic PLH might be pregnant without any risk of thrombosis for her or for the kids. So really, it improved a lot. However, we found some limitations with eculizumab, and especially coming back to normal hemoglobin level. So what we did with a couple of friends and colleagues from Brazil, Italy, we did decide to try to see if we could, in a way, categorize the response to eculizumab in our patients. So we were able to reach 160 patients that were all treated with eculizumab. I'm sorry, there is still some French here, but anyway, you will have a French lessons at the same time. So it's important to see that the median duration of the treatment was almost 6 years. So it's really a real well done reflect of a real life of patients under eculizumab. Of course, we censored when the patients were included in trials, 35, or were transplanted. There was 2 patients who were transplanted in the cohort of patients. For the dosage, as you can see, the repartition is almost what we are doing. Most of them were treated with eculizumab 900 milligram, while 20% roughly were treated with 1200 milligram. And what we found in that study is that if you look for hemoglobin levels, this is on the right side of the screen, you can see that there are some patients who are able to reach more than 12, between 10 or 12 patients who are not regular transfused, but are between 8 and 10, and finally patients where it doesn't change anything. And as you can see, there was a bunch of patients that were not optimal responders, because so we call this complete response, good response, partial response, minor response, and as you can see, 20% of the patients, a little bit more, were in complete response, while all the other one could have been benefiting from another treatment. And in the end, you see that almost 60% of the patients are well controlled and are not transfused regularly. On the other way around, you have almost 30% of the patients that are still transfused or need a transfusion once a while. So you see that basically eculizumab improve overall survival, decrease transfusion rates, decrease thrombosis occurrence, making patients coming back to a, I would say, quite normal life. But however, there are still some unmet need in terms of hemoglobin level, quality of life, and come back to strict normal hematological level. So of course, we look also on different time period, because I told you we have 6 years follow-up. And it was quite basically the same. I mean, if you look for the first 6 months and then the 12 months, and you see that you're still some patients that are improving with time. This is also something that is outstandingly important, is that in a way you need to wait for eculizumab to be efficient. And you see that you're reducing the number of patients with partial or minor response because you have an improvement with time. But anyway, when you look in the last 6 months after treatment, you see that some of the patients are still requiring something else. So this is the complement cascade. This is quite difficult, I agree. I mean, there is 2 things that we are always frightened of in medicine. This is the complement cascade and the coagulation cascade. So I'm not going to enter into detail. The idea of this morning is clearly not to make it complicated more, it's just to simplify the system. So if you want to simplify, there is 2 ways to block this complement cascade, or the terminal blockage. So just before the MAC is forming. So basically, just before that, your hemolysis the cells, or, and I'm sorry, it's not on that slide, but you will see it later, or the proximal complement blockers, blocking C3, factor B or factor D, where we are blocking very early the complement activation. And that is something that is different. You are blocking, and that's what is coming at the moment in the field. But I could not speak about the other ones because they are also in the market. But of course, we are more interesting with the complement proximal blockers. So first of all, let's come back to what is more or less eculizumab, so ravulizumab and crovalimab. So again, they are blocking at the C5 level, so they are terminal complement blockers. The only advantage, but it's important for patients, is that it can be injected every 2 months. So ravulizumab is eculizumab, not more, not less. The only difference is that it has been modified on amino acids. 2 amino acids has been modified and are giving a kind of cycling period of times that you are injecting them every 2 months. So you can see there was 2 trials that's been published in Blood in 2019, the 301 and the 302. The 301 was patients who have never received complement blockers. This was a naive study, and as you can see, they were randomized to receive ravulizumab with a upload dose day 1, and then a maintenance dose from day 15, and then every 2 months. And on the other way around, you have eculizumab with a 4 weeks induction period of time, and then the maintenance period for the week after. So you can see that the primary endpoint were at 26 weeks, and then afterwards, all patients, and that was the advantage to participate to those trials, were able to receive the maintenance dose of ravulizumab. They were all treating with ravulizumab, and that was quite important because then you have access to the best treatment. For the 302 study, there was some patients who were already exposed to eculizumab, and they were switched to ravulizumab randomly or to continue eculizumab. So as you can see, again, randomization, and then or ravulizumab or a standard treatment with eculizumab. And again, all patients, including them who have been randomly assigned to eculizumab first, were able to receive ravulizumab afterwards. So those are the primary endpoints of those studies. The 301, I remember to you, this is naive patients. So the primary endpoint was transfusion avoidance to comparison with eculizumab, and LDH normalization. And for the 302, since they were already under complement blockers, endpoint was the percentage of change in LDH from baseline. The secondary endpoints were quite classical in PNH with a FACIT score, breakthrough hemolysis, hemoglobin stabilizations for both studies. And there was also PK/PD endpoints that has been published in the British Journal. I didn't have time to show you anything, but you can refer to it if you are interested in. So those are the 2 populations who have been assigned to those studies. There was more than, a little bit more than 200 patients in the 301 study and 200 patients in the 302 study. All the parameters were quite equilibrated. There was the same number of patients in each group. The only thing that I would like to remind is that if you see for the 301 study, there was 60%, almost 60% of patients coming from Asia, while for the 302 studies, those patients were mainly coming from Europe and North America. So in other words, just to know that, of course, the drugs are developed in countries where the drugs is needed. So when you are checking for, when you're doing a trial on naive patients, of course, naive patients are not anymore in Europe and North America. And so, of course, Alexion went to Asia to include those patients. That might be a problem when you're going to register the drugs, because there is some genetic factors that make that the drugs might not be responding the same. But anyway, there was also 30% of patients coming from with a Caucasian background, so that was enough for regulatory agencies to say that it's okay to have a market approval. There was the only differences between both studies. In terms of primary endpoint, as you can see, again, naive patients on the left, you can see that the bar is still crossed, so there is no difference. There was, and I should have been begin by this non-inferiority trials, and you can see that there was non-inferiority in terms of transfusion independence and LDH normalization. On the other way around, you see that for the 302 studies, there was almost a difference in terms of LDH percentage changes that will favor ravulizumab. However, again, the design was done for non-inferiority. So even if this bar has been crossed, it would have changed anything since the design of the study was not to show a superiority. So basically to say, ravulizumab is exactly the same than eculizumab. I should say confidentially that ravulizumab have been developed like eculizumab 1200. This is the reason why the results might appear a little bit better. But in the end, this is exactly the same. And on the long term, it has been shown that ravulizumab is strictly identical to eculizumab, and that the only differences were in terms of injection every 2 months, so only 6 times per year, compared with every 2 weeks, so about 26 times per year. So this is not nothing. This is a huge improvement in healthcare system. That's the reason why in France it has been granted and scored quite highly, even if in the end it won't change anything in terms of response for our patients. So now, another one that is still targeting the C5 and terminal blockage, it's C5 by crovalimab. This is a drug that has been developed by Roche, and that is still in development. I will speak later on drugs that are in development at the moment. So these drugs is really a smart drugs, and they are calling this the smart process. Basically, as you can see here, while in the circulation, the drugs is with high affinity with C5, she's reaching a target, and then she's opsonized. And when she's opsonized in the endosomes and lysosomes, the pH is acid. And so at that time, there is a separation with the C5 that goes to the lysosome and are destroyed. But the antibody has also been engineered with a FcRn receptor that is of high affinity in acid conditions to the endosomes, and then is staying in the endosomes while the target is degraded. And it's created a new circulation of the antibody. So you see how smart it is. You have a high affinity that is not the case in acid conditions. So the C5 is degraded by the lysosome because there is a separation in the endosomes, while the FcRn receptor is protecting the antibody from degradation and making him recirculating. And it's like this. So that's very smart. And it's given every month. It's a sub-Q injection. And basically, that's what has been published. It was able to control the complement activity. We know that the complement activity is never controlled the way it's controlled in healthy volunteers. But however, you see that the C5 concentration in eculizumab and treatment-naive patients were in a row of what has been published already by patients under eculizumab. So in other words, PK/PD showed that this drug was able to control and to block the complement in satisfactory conditions in patients with eculizumab that has been switched from eculizumab to crovalimab, or in patients who have never received complement blockers who are naive to a PNH treatment. So to make a long story short, what has been shown is that crovalimab is able to achieve terminal complement inhibition in patients with PNH. It is well tolerated. There was no AEs. That was important with these drugs. The efficacy was the case for most of the patients. The breakthrough hemolysis events were not frequent, and basically these drugs is at the stage of phase 3 worldwide. Those trials are called Commodore 1, Commodore 2, and they are still ongoing at the moment with accrual, and they are checking crovalimab in a randomly way to eculizumab or ravulizumab for naive patients, and the same for switch patients. So I would say this is the new drugs that have been developed for the terminal blockage. Let's now enter in the new drugs that are a proximal blockers. And the three main drugs that are at the moment in the field with burning issues and revolutionize the field is IPL2, pegcetacoplan. This is an anti-C3 target drugs. Factor B, LNP023. This is the iptacopan. So here it's Apellis, and Sobi in Europe. Here it's Novartis, and finally you have Alexion, who developed danicopan. It's an anti-factor D, and honestly, it was before Achillion and then Alexion, that is now an AstraZeneca company, bought the danicopan. So you see how complicated it is, because you have to follow the scientific flow, but also the company modifications, because it's also modified the way of developing the drugs. For example, the danicopan at the moment is only used in combo with eculizumab or ravulizumab, while the other ones are in monotherapy. So the most, I would say, burning topic at the moment is the pegcetacoplan, an anti-factor 3. It's a sub-Q injection 2 to 3 times per week that has been published in March this year. And basically, and that is outstandingly important, just something to remind to you what is going on with eculizumab. When you have the activation, I told you, there is an activation from the beginning. The C3 is going on the red cells, as you can see here, under eculizumab. And then eculizumab is blocking the complement activation at that phase. But eculizumab cannot avoid C3 to stick on the membrane of the red cells. And under eculizumab, this is responsible for what we call extravascular hemolysis. So in other words, the idea was that the unmet need with eculizumab, showing patients remaining anemic, might come from the fact that 30% almost of the red cells are opsonized with a C3 deposition, and then that might be responsible from an extravascular hemolysis. On the other way around, if you are using pegcetacoplan that is blocking very early the complement, you can see that there is no deposition. There is no deposition because it's already blocked. So there is no reason that the cells were depositing on the membrane surface. And then, of course, you are blocking the extravascular hemolysis. So in other words, pegcetacoplan is able to block the intravascular hemolysis, like eculizumab, but it's also able to block the extravascular hemolysis, as you can see here. So this is, I think, the message, the most important of my talk, because this is this way that the proximal blockers are working. And if you take the factor D or factor B, this is exactly the same. They are very proximal, so they are avoiding the deposition of C3 at the membrane cell surface, and they are avoiding extravascular hemolysis while protecting, like eculizumab, for intravascular hemolysis. So this is a New England paper. It has been shown that the design was a run-in period with the two drugs, eculizumab plus pegcetacoplan, and then they were randomly assigned to pegcetacoplan alone or eculizumab, and at the end, again, there was an extension phase. So I told you this was targeting patients who were suboptimal responders to eculizumab, so they have to be adults, because running trials in pediatrics, even if, of course, this is worthwhile, it's quite more difficult. And there was for patients who were not optimal responders, so with an hemoglobin level of less than 10.5, they were anemic, highly anemic. And the other things I told you, they have to be presented extravascular hemolysis. And if you have extravascular hemolysis, you are regenerating because you have hemolysis. And that's the reason why the reticulocyte counts have to be more than 100 giga per liter. And that's, of course, also avoid patients with bone marrow failure. And as you can see, this is the reason why there was some exclusion criteria for patients with less than 50,000 platelets, less than 500 neutrophils. So again, the distribution of the population, you can see, was about 80 patients, and they were well distributed with time. They were well distributed according to transfusion dependence, and also according to race. And basically what this study showed, even if the primary endpoint was about 16 weeks, it showed that there was a drastic improvement in terms of hemoglobin levels. So I'm sorry, because there is missing something here. There was 4 gram hemoglobin differences, 3.8 to be precise. And it was, of course, highly significant. And as you can see here, the patients who were anemic under eculizumab in the running period, they were, of course, gaining from the combo, and then you were stopping one of the drugs if it was eculizumab. And the patients staying under pegcetacoplan, the hemoglobin level was sustained, stable, and close to normal. While if the patients was assigned to eculizumab again, then the hemolysis was going on and the hemoglobin level was reaching 8.6. This is what it was before inclusion. So, of course, the primary endpoint is a good thing, but it's even better when the secondary endpoints are showing drastic improvement. So basically, in terms of transfusion independence, there was a superiority for pegcetacoplan, showing that about 85% of the patients at the end were not transfused anymore. You were normalizing reticulocytes using pegcetacoplan because you were controlling the extravascular hemolysis, so there was not any more reticulocytes elevated. The ALDH were not changed, which is outstandingly important because it's good to control extravascular hemolysis, but it's also good to stay doing the same things that eculizumab was doing, especially in thrombosis occurrence. And of course, if you are improving hemoglobin level, if you are not transfused anymore, if you are controlling the extravascular hemolysis, you are improving quality of life. So this study was quite striking because for the first time, patients who were not optimal responders to eculizumab were able to improve hemoglobin level and quality of life. So something that is not published yet, this is the reason why I did not send to you my slides, because there is many data that are not published, that I presented to EHS this year was a long-term of those patients treated by pegcetacoplan. So the New England paper is about those 16 weeks first treatment period. And on the other side, you have the 32 weeks long-term follow-up. So you have a 48 weeks data here with all patients treated with pegcetacoplan. Again, this is exactly the same inclusion criteria. It's just a longer follow-up of patients who are under pegcetacoplan because of being suboptimal responders to eculizumab. And what we saw is that for the great majority of the patients, so this is what I showed you before in the 16 weeks period in the New England paper, you see that the hemoglobin level for patients who were randomly assigned first row to eculizumab but then switched to pegcetacoplan were also very good, with patients switching almost normal hemoglobin level and being free from transfusion. So it's a good news. The other good news is that if you look for LDH in retics, you can see that the results with the extended period of time was exactly the same than the one that we observed at 16 weeks. It's mainly normalization of LDH and normalization of retics. And that is also something that is good. And the other thing is the being free from transfusion and the improvement in terms of quality of life. You see that, as I told you, there was about 70%. You see here patients who were continuing pegcetacoplan, so they were first of all randomly assigned to pegcetacoplan and continued pegcetacoplan, and at 848 there was 73% of patients transfusion independent. And for the patients who were under EQ, you see that there was 15% at 16 weeks because they were under eculizumab. They were switched to pegcetacoplan in the extension study, and you see that at week 48 there was 72% of patients transfusion independent, and that was reaching an improvement in terms of quality of life highlighted by the fatigue facet score. This slide is outstandingly important because it's showing that while we are improving hemoglobin level, we are decreasing transfusion dependency, we are also making the PNH cells disappearing from C3 loading. So I told you extravascular hemolysis is just a physiopathological consequence of eculizumab. Eculizumab is blocking at the C5 level, so it's not avoiding C3 deposition, while when you are blocking the proximal levels, you're avoiding this C3 deposition. And you can see here nicely the C3 deposition on red blood cells you see that when you are under pegcetacoplan, in the first phase, you have no cells or very few that are C3 loaded, while under eculizumab, the cells are still loaded with C3. And when you are switched to pegcetacoplan, then you have a drastic decrease of C3, and you have no C3 levels anymore. Of course, there is another consequences of this, and that is what we have to keep in mind, is that almost all cells became PNH. So in other words, since you're not destroying PNH in the intravascular hemolysis, and you are protecting them from intravascular hemolysis and protecting them from extravascular hemolysis, we saw an increase in terms of clone size. And this is the main message here. As you can see, for patients who came from pegcetacoplan to pegcetacoplan, there was 90% clone size in the blood. So the hematopoiesis was almost completely reliable on PNH. And when you switch from EQ to pegcetacoplan, again, 60% of PNH clone size for patients under EQ at week 16, whereas they are switched to pegcetacoplan, this is about 93%. So the main, I would not say problem, but the main fear of that proximal blockers is to make the blood almost only PNH. And what's going on when you make blood almost only from PNH? That you're exposing your patients to more severe breakthrough hemolysis. And that was also the message from that study, showing that 13 patients stopped pegcetacoplan because of many reasons. There are some reasons that we know might be related to the disease, acute myeloid leukemia. We know that PNH is a disease that might evolve to acute myeloid leukemia or MDS or plastic anemia, so this is not surprising. There was a COVID-19 infection in one of my patients. That was, of course, the pandemic reason. The pancytopenia, again, in bone marrow ferrous, is something that is, in a way, predicted. However, as you can see, there was also breakthrough hemolysis for 6 patients. 1, 2, 3, 4, 5, and I don't know where is, and 6. So you see that it was you have to stop the drugs, and there was also a mesenteric ischemia that, of course, we don't want to see in patients under blockers. It was also related to a breakthrough. So basically, what's shown here is that it's new drugs, it's outstanding for the majority of the patients, but you have 15% of the patients that might be exposed to higher rates of a breakthrough, severe breakthrough. And that's what we are at the moment trying to understand, if it's PK/PD problems, if we should inject more patients, if we should re-inject eculizumab at the time it's going to be the case. And that was also, and you'll see a discussion in the New England Journal that should come soon on that particular problem. So now the second drug that is outstandingly important as a proximal blockers is LNP023, iptacopan, anti-factor B. Those are the in vitro studies that have shown how efficient it is. So iptacopan is a proximal blockers, as I told you, and as you can see here, you see that the hemolysis of red blood cells in vitro studies were very, very well controlled by anti-factor B and anti-factor D inhibitors, while under anti-C5, you have a control was less. I was also showing that the red cells were protected, intravascularly speaking. And as you can see here, this is outstandingly important. This is some in vitro inactivated acidified serum, showing that if you have patients that have a PNH, you have normal cells that are CD59 positive, and you have a PNH cell CD55 negative. You know that it's a GPI negative anchor proteins. As you can see, if you don't use any complement blockers, all the PNH cells are destroyed. So this is shown here. If you are using eculizumab, you see, as I was telling to you, both populations. You have, first of all, normal populations, GPI anchored positive, but you have also GPI anchored negative cells with cells that are C3 quoted here. And that is responsible from extravascular hemolysis, and that's what I was repeating during the talks. However, when you give anti-factor B, but it's true with anti-factor D, and it's true with the drugs I showed you before, anti-C3, you don't have any more extravascular hemolysis because you don't have any more C3 deposition, right? So this is a phase 2 study that has been published in the Lancet. We were doing with Antonio Risitano and myself. Basically, we were also targeting patients with a non-response to eculizumab. There was 10 patients, all were transfused under eculizumab, and the primary objective was to see if we were able to reduce the extravascular hemolysis, and of course to see if the drugs was well tolerated in those patients. And what we did, I'm sorry to come back to this, but what we did is that we used different dosage of iptacopan, always with eculizumab, and at a time when even it's not shown on that slides, we decided, because of the outstanding results, to stop eculizumab in the patients who are receiving anti-factor B in monotherapy. So we included 10 patients. So it was France, Germany, and Italy. All patients were transfused before inclusion and treated with eculizumab or only eculizumab with 900 or 1200 milligram every 2 weeks, and the patients were transfused while under those drugs. And basically, we gave all patients iptacopan plus eculizumab. And as you can see here, while patients were, for some of them, showing LDH elevation, you can see that under the drugs, the LDH were outstandingly well controlled. If you look for hemoglobin level, I remind to you that all patients were transfused before. As you can see here, all patients on the right side were not transfused anymore under combo, and the hemoglobin level, as you can see here, was almost reaching the normal for most of the patients. You see there was some patients that were not normal, but was reaching almost normal. The other thing, of course, that is important, that the 3-3 opsonization disappeared because it's mainly controlling intravascular and extravascular hemolysis, as I told you. And as I told you before, you see this increase of the PNH clone size in the blood, since you are protecting the cells from intravascular hemolysis and extravascular hemolysis, then the blood is almost, hematopoiesis is almost PNH related. So that study was nicely published at a high level. It was showing that these drugs is able to control PNH. There was improvement in all the parameters. And the other thing, as you can see, the most important is what is written here, is that all the patients were retaining a benefit in monotherapy. So for some 7 patients, but now it's 10, who stopped the drugs, the stop eculizumab, the hematological response was sustained with time and was able to show a drastic improvement compared to baseline. So the interest of that drugs is that it's an oral compound. So basically, we don't have IV or sub-Q injections anymore. It's just a pill that you're taking twice a day, morning and night. So, I mean, this is really a revolution in the field. So just a word on the studies that are ongoing at the moment. You have APPLY studies, that is patients who are still under eculizumab but are not having a outstanding response, and the APPOINT PNH study, that is patients who are naive to complement inhibitors, all receiving iptacopan monotherapy. So follow please those studies, because once they will be done, it will probably drastically change the PNH field. So the last one, I mean the 2 last one, but the one that I'm going to spend 2-3 minutes is the anti-factor D from Alexion, but at the moment it was Achillion. This is the reason why it's ACH 4471. It's called danicopan, and of course you're gonna heard about it because this is an oral compound too. I'm not gonna enter into details because of matter of time, but what I can tell you, it's exactly the same than the one I presented to you before. It's acting at the same level. It has been developed the same way. The only thing that we are missing is data on monotherapy. So again, I told you, Achillion developed the drugs, was bought by Alexion, and in a way, in the first phase of development, Alexion associated it with ravulizumab and eculizumab as a combo that, in a way, I think for patients is not the best way to treat, because if you're receiving 2 complement blockers, you are just burning the way of driving those drugs, and it's not really improving anything for patients. So now it's gonna come on a monotherapy development, but at the moment it's combo. So as you can see here, those are the patients who were included in a combo study. They were all transfused with red blood cells. And then after treatment, you see that there was only one transfusion. I told you this is exactly the same results than the anti-factor B. And as you can see, it was decreasing drastically the transfusion in terms of annualized event rates or a number of transfusions. Of course, if you're not transfused anymore, if you're blocking intravascular hemolysis, your hemoglobin level is increasing. And as you can see, reaching almost normal level, even if it's less impressive than what has been published with the anti-factor B. Again, I told you, and I should present this one first, is that you see that basically the C3 deposition under treatment is decreasing. That's what I was telling to you. You're protecting the cells because there is no C3 deposition because you're blocking earlier. And if you're doing this, the clone size is increasing. So basically, this is very important. You're protecting from extravascular hemolysis. You don't have any C3 deposition anymore. Your patient is not transfused anymore, but the clone size is outstandingly high. So if there is a breakthrough hemolysis, you may be exposed to a severe breakthrough, higher severe breakthrough than the one you will be exposed under eculizumab, because again, under eculizumab, we are half 60-40% of normal hematopoiesis and PNH. And to be complete, I'd like to just say a word on this one, but there is, yeah, sorry. So first, there is a randomized trial going on with danicopan, but the last one I would like to speak is the anti-factor B from a company, a small company. It's an Australian company that is called BioChrist, and which is developing an anti-factor D that is exactly the same than danicopan, BCX9930. I'm just quoting the studies because the data are exactly the same, and I don't want to overburden you with all those data, but it was important just to hear because they are going to come to you as they are coming to us to develop their drugs. So this one for switch, this one for naive. So this is my conclusion. I will probably a little bit longer than expected, but we are really facing a revolution in the field. Again, we are coming from a standard of care with eculizumab that improves survival, that did reduce transfusion dependency, that improve quality of life, protecting from thrombosis, but with unmet need. I show you 60% of patients who are still anemic or transfused once a while. And on the other way around, we have some studies coming in with anti-C3, anti-factor B2, anti-factor D, with great improvement in terms of hemoglobin response, but also the fear that those patients might be exposed to higher rates of hemolysis in terms of breakthrough. Very early studies, only one long-term study, anti-C3, the other one is still in phase 3, so we have to be very cautious. But anyway, I think this is fascinating, and this is really a drastic changes, not only for PNH, but also for all the diseases that will be complement-related. Last but not least, I'd like to thank all my colleagues at the local level in St. Louis Hospital who are working with me on this fascinating disease, and at the international level, of course, Austin and Antonio, with whom I'm always interacting, exchanging, and discussing those drugs. Thank you very much, and I will be more than happy to take your questions.
Thank you, Professor Lecter, for updating us with so many advances taking place and so many breakthroughs in the treatment of PNH. I only hope that sooner or later they come to our patient level. But fantastic, what a review and what an update. So we have our Indian faculty. They would like to ask you some questions. Dr. Venkatesh.
Yeah, thanks again, sir. Thanks for the small French lessons, Professor Regis. What I would like to ask you is, does breakthrough thrombosis even occur with these early and late complement inhibitors? Because none of your secondary endpoints mention about thrombosis.
Yes, this is a very important question. I think that we have tendencies to forget thrombosis, which is the main prognostic factors in PNH. For the moment, the thrombosis we observed with the complement blockers we have in ENS was really at time of breakthrough. It was not when patients were controlled. So I think that there is two parts of the answers. The first one is that when the patient is doing great, and it's about 80% of the patients for the anti-C3, for example, the thrombosis occurrence is zero. It's like eculizumab. It's exactly working well. But in terms of breakthroughs, then you are facing problems. That's the reason why at the moment we have those discussions with Pete Hillman and others. And we are really thinking that if you are exposing patients, I mean, if you are facing a very severe breakthrough hemolysis in patients under proximal blockers, you will probably have at least temporary to inject again anti-C5 because then you see that the drugs is completely overloaded. And we didn't have so much this feeling with anti-factor B, but anti-C3, there will be probably the case to inject anti-C5 plus higher dosage and more dose short interval of anti-C3 and then coming back to a normal phase.
Thank you.
Thank you. Dr. Pooja.
Thank you, Dr. Regis. In continuation of the previous question, are you using anticoagulation concomitantly with these newer drugs?
So in PNH, we did a study that we published in Blood in 2008. It was a French registry. And what we did observe is that the anticoagulation was not able to avoid the thrombosis occurrence. And when the patients were under anticoagulation, they were exposed to a risk of hemorrhage. So that's the reason why we are not advising using anticoagulation as a primary prophylaxis when the patients are under complement blockers. So at the moment, we have exactly the same guidelines for proximal blockers in terms of, I would say, patients that are at equilibrium, that are not presenting any problems. And then if a thrombosis is occurring, then yes, we are advising to continuing anticoagulation lifelong to avoid a secondary thrombosis, because we know that the main risk, whatever the thrombosis disease is, of a thrombosis reoccurrence is having already been exposed to a thrombosis. So not use anticoagulation as primary prophylaxis while you are under complement blockers, but use, of course, lifelong with complement blockers if you have experienced this event, because it's devastating in our patients.
Thank you, sir.
Thank you. Dr. Mona.
Yes, sir. So my question is, sir, as eculizumab is not available with us, what are the indications for starting primary thromboprophylaxis in PNH patients? And is there a role, is there a cutoff for the PNH clone size?
You mean warfarin?
Yes, sir.
No, we are not using anticoagulation. Neither warfarin, neither heparin, low-weight heparin, or whatever. Again, the risk of hemorrhage is higher than the prophylaxis you may have in those patients because they are going to be exposed to the risk. And we showed, basically, the study was quite nice. It was while we didn't have access to eculizumab. So patients were receiving just supportive care, was before 2008. And general practitioner, as you are thinking now, were proposing anticoagulation primary prophylaxis for PNH patients with hemolysis, thinking that they will be protected. And basically, in that study, the patients who were receiving warfarin were highly exposed, significantly speaking, to thrombosis. Not because warfarin was a risk of thrombosis, but just because a general practitioner in a core of PNH patients identified the patients who are high risk with hemolysis, placed them on warfarin, but it did not avoid a thrombosis occurrence. So we are not advising primary prophylaxis with anticoagulation. However, on the other way around, because the disease is very severe, what we are advising is that for patients with hemolysis, high-rate hemolysis, transfuse, and so on, you might discuss and still discuss bone marrow transplantation. Because when you have siblings, it's better to have an 85% overall survival. It's what we have with PNH patients outside eculizumab when they're only hemolytic and not thrombotic and not aplastic. We have an 85 overall survival. Of course, there will be GVHD for some of the patients, but they will be alive. And basically what you have to know is that the median overall survival time for patients with PNH with hemolysis is 20 years. So basically, if you're not giving a treatment, you're going to lose some patients with years. So that might be also something to discuss. And yeah, it's a bit, speaking about transplantation now, it looks like sometimes a bit surprising, but no, I mean, that's what we are also advising to our North African colleagues that have access to transplant, but not to eculizumab on a regular basis. And they are transplanted some patients, young patients with hemolytic PNH because they have many siblings, and that might be also a good option.
Thank you, sir.
Dr. Rima.
So is there any cutoff for clone size, and is the exact clone size important to determine the treatment strategy and predict outcomes?
Yeah, for the predictors of outcomes, patients who have a clone size higher than 30% are exposed to higher rates of thrombosis. But what we will show is that all patients with a PNH clone can have thrombosis. So basically, if a patient has a bone marrow failure, is has a PNH without any hemolysis, it might be also exposed to a risk of thrombosis. So the message here is that all patients with PNH might be exposed to thrombosis, and the risk is higher while your clone is hiring 30%, and 50% is also a clone size that is related to thrombosis. For the strategy, we are not taking into account clone size to introduce treatment because they are so costly that. So for us, we are using complement blockers if the patients have intravascular signs, clinical signs of intravascular hemolysis, abdominal pain, fatigue, and so on. And sometimes it's really correlated with the clone size. And also when the patient is transfused because of hemolysis reason, and of course if they are presenting thrombosis. And we are also giving eculizumab on a regular basis to pregnant women to avoid thrombosis at delivery. So those are the four, I would say, factors that make us prescribe eculizumab. You see far from the biology in the end, even if, of course, someone with high rates LDH and high clone size is more able to receive the drugs, but he has to present clinical signs or be transfused because, I mean, we need some good reason to pay the price.
Sir, any significance of chasing PNH in MDS patients?
You mean? I don't understand the question.
PNH clone, is there any need to follow up? Is any significance of PNH
Yes.
in MDS?
Yeah. So for patients with PNH, even if they are only hemolytic, it's a bone marrow failure disease. So you have to do some marrow sampling every 12 or 18 months. We have a clonal evolution rates of 10 to 15%, 5 to 10 years. And of course, you don't have PNH without without signs of bone marrow failure, even if it's not something significant clinically speaking. If you're not seeing in the periphery, in the blood of those patients, if you do marrow sampling, if you do biopsy to those patients, you'll see that the cellularity will be lower. So it's very important to follow those patients because then they are exposed to genetic instability like bone marrow failure patients. So we are doing marrow sampling every 18 months approximately. Because it's far more easy to transplant a patient with only MDS at the clonal evolution, cytogenetic speaking, while when they are morphologically transformed.
Thank you, sir.
Thank you. Dr. Pooja.
Sir, I have two questions regarding management of PNH in pregnancy. Do we need to monitor the clone size? Is there any significance of monitoring the clone size? And also, is there any significance of monitoring the clone size in an overlap aplastic anemia PNH patient who has a very small clone and is being treated on immunosuppressive therapy? Do we need to monitor the clone?
So for pregnancy, we are more looking on LDH. What we do know is that the risk of thrombosis is when patients have 2 times LDH or more. So what we are doing is really looking on this, and most of the time it's associated with clone size that are higher than 30%. So we are looking on LDH, and if the patient is hemolytic, we have no restriction. We are prescribing eculizumab, 900 at the very beginning, first 6 months, and then 1200 in the last 3 months, because there is an increase in terms of vascular circulation. There is a 30% higher because of pregnancy. So we are increasing the dose, and we are coming back to regular dose H900 at time of delivery. So this is for pregnancy. For patients with bone marrow failure and PNH, I heard a question at the very beginning saying that is it something related to acquired aplastic anemia? The answer is yes. to have an expansion of PNH cells, you have to present behind an idiopathic, and idiopathic means autoimmune mediated bone marrow failure. So this is helpful in the pathophysiology. It's just saying to you that this patient is presenting an acquired disorder, and sometimes, especially in adolescents, young adolescents, it's something helpful because you have a PNH clone and then you have an acquired disorder. And if the clone size is low, we know that 20% of those patients with bone marrow failure and PNH might evolve to hemolytic PNH with high clone size. So we are advising to do a PNH clone every year to see what's going on, but most importantly, to follow those patients clinically, to ask them if they have abdominal pain or if they have something that is different than usual, to detect the hemolysis behind that might be a factor of treatment. But to answer properly to your question, it's once a year.
Thank you. Dr. Prusti?
Yes, sir. Thank you, sir. Dr. Regis, there is a question that how should monitor the ecology of dosing schedule, or is there a mandatory to determine the serum levels to monitor the effects?
Yeah, so the approved drugs is 600 every 4 weeks and then 900 every 2 weeks. So we are always beginning with this. And then if the patient is not an optimal responder, we are first checking for weight, because we know that we have some good results like 1200 with overweight patients. So we are looking at that time for eculizumab level and CH50 blockage. And if the patients have not a complement that is well properly blocked, or if the eculizumab level is not in adequate range, then we're increasing to 1200. The thing that is important, I should have begin with this, is that we are not modifying anything before 6 months, because I showed you at the very beginning of my talk, there is an improvement with time. So we are really waiting 6 months, and then we are checking for this to see if the complement is well blocked and if the eculizumab level is okay, and then we might increase to 1200. Then we came to the other situation where eculizumab might be not so efficient. If there is a bone marrow failure, this is why it's so important to check the marrow regularly to avoid bone marrow failure associated syndromes. And then we are going for infections. We know that if there is a infections, a flu or something else, that might also be tricky, because this is consuming and secreting complement as inflammation. And so it's of course difficult for the drugs to block if there is an inflammation going on at the same time. And then afterwards, we are checking for extravascular hemolysis to see if there is a C3 deposition in cells that might benefit from proximal blockers. And also, but more in an Asian background, of the C5 polymorphism that has been published by Nishimura in the New England, I think 5 or 6 years ago. But we did not have those kind of patients here.
Yeah, thank you.
Dr. Aditi?
Thank you, sir. So my question is there any increased risk of infections with the proximal complement inhibitors compared to eculizumab?
Yes, theoretically, yes. So basically, if we are blocking at the C3 level, we are not opsonizing anymore the cells. While when you are blocking at the C5 level, you're still having this opsonization still going on, which is limited the effect of eculizumab, but at least which is protecting against some of the infection, especially pneumococci infections and Haemophilus influenza. So yes, theoretically speaking, there is higher rates of infections, but for the moment, clinically speaking, we did not observe this. We are giving vaccines to our patients for meningococci, all serotypes, including B, but also Haemophilus influenza and pneumococcus. And we're giving, at least in France, primary prophylaxis with antibiotics, with penicillin, to avoid this risk. So I would say for the moment, we didn't see any red signals, but it has to be monitored, followed with time, because I guess that when the drug is going to enter into a daily practice, it should be probably a risk. So the answer is theoretically yes, practically no, but long-term follow-up requested and mandatory.
Thank you, sir. So I have one more question. Since eculizumab is not available and affordable right now in a patient with hemolytic variant and thrombosis, what treatment options can we offer?
So as I was telling to you, if there is no thrombosis, transplantation is a good one with siblings. If the patient has already presented thrombosis, I have no option. Why? Because we did retrospectively study 200 patients with PNH in Europe who were receiving transplantation because of or hemolytic or thrombosis or plastics. For hemolytic, the results are quite good and acceptable. We don't have access to eculizumab. But when patients have already presented a thrombosis complication, the results were awful. The overall survival was 50%. That means that 50% of the patients died because of the transplantation itself. So in this way, we are advising to really well anticoagulate those patients and to try to have a compassionate use of complement blockers, but there is no option. I mean, transplantation is really too high. The risk of transplantation is really, really too high to propose transplantation to those patients.
Thank you, sir.
Dr. Akshaya.
Thank you, sir. So my question is, how do you suspect eculizumab resistance?
So the first change will be the switch to ravulizumab. And all patients are going to switch to ravulizumab, I think, quite quickly. And then afterwards, as I was telling to you, the phase 3 study are almost available. Sorry, my kids are coming. So the proximal blockers won't be available soon because, again, the phase 3 trial is still ongoing. So we just have the results from the anti-C3 treatment, and the other ones are ongoing at the phase 3 level for the anti-factor B, factor D, BioChrist, or Achillion. So we need the long-term follow-up of those studies. So it's going to take time. So all this to say that I think ravulizumab is going to come quickly to the field next year, at least in France. We don't have access to it. We'll have access next year, I think. For the other ones, I mean, with all the regulations needed, the studies, the long-term follow-up, the data generation, I mean, we need at least 3 or 4 years before being available regularly.
Sir, how will you clinically suspect that the resistance is developing against eculizumab?
Sorry, repeat the question.
Sir, how to suspect that eculizumab resistance is being developed?
So for me, there is no resistance to eculizumab. We cannot speak about resistance. There is some patients who are responded to it and others that are less responding to it, but for many reasons. There is no, it's not like antibiotics. You're giving antibiotics, there is a resistance, and then you are selecting germs, and then that's, and so on. No, eculizumab for a patient is working or not working. Of course, we are checking genetic background and hereditary disorders, some things that might probably affect the response to eculizumab. But it's really black and white. It's not something they're acquiring with time. So then what we have to dissect, and that I was telling to you before, according to weight and C5 polymorphism, bone marrow failure disorders behind, and so on, the patients who are less responding to it. But we don't have yet a clues on resistance. I mean, the patients are responding or not responding. Of course, no, just to complete. In time, I mean, during evolution, some of patients could present MDS, AML, or plastic anemia. So in a way, it's not resistance, it's just because the disease is changing. And then, of course, there are going to be lower responders, but just because the disease is changing. That's the reason why it's very important to monitor those patients on a regular follow-up.
One question from the audience. While working up a patient of aplastic anemia, if you find a significant PNH clone, but there is no hemolysis or any other complication, does it impact your treatment?
No, the response is no. We cannot afford to treat all patients with high clone size because of the price of the drugs, and because, again, most of the time, the clinical consequences are when patients are suffering from symptoms of intravascular hemolysis. So again, if there is a high clone size, of course, we're checking for clinical signs, fatigue, abdominal pain, nausea, stuff like this. And if there is nothing of this, we are not taking care about the clones. Of course, we are repeating it to see what's going on, but we are not changing strategy. And this is the same for bone marrow failure. If you have a patient with acquired bone marrow failure and a PNH, if there is no hemolysis on the PNH sites, I mean, please do the same than you will do with acquired bone marrow failure without the PNH clone.
Right, sir. Another question is, after anti-factor B use, if the PNH clone size is increasing, does it create a clinical problem?
It's clear. It's clearly not a clinical problem per se, but it's a clinical not prediction, but it's a possible risk of severe hemolysis. So in other words, if you are creating PNH hematopoiesis only, it means that if for a reason you have an hemolysis, it's going to be terrific. It's terrible, because you have no protection for the normal hematopoiesis so far. So that's the reason why you're exposing your patients to a very severe acute breakthrough hemolysis that might them even at life-threatening conditions. That's what we observed with some of the patients with proximal blockers. I mean, they were giving some infections, and then they were destroying all the PNH cells. And so we were coming from 12 gram hemoglobin to 4. And that is a major and huge problem. So it's not something creating clinical problem per se. We have 80% of the population with proximal blockers are living well. They have no problem with this, but might be difficult for 20% who are exposed and suffering from very severe acute breakthrough hemolysis.
Right, sir. Another question is, with using these agents, do you give prophylactic antimicrobial agents?
Yes, we are always giving penicillin to our patients. This is very debating at the international level. Some of the experts are giving only vaccination. The pharma companies at the moment for terminal blockers are also advising vaccination only. The only thing is that I have some difficulty, theoretically speaking, to think that the patient, even vaccinated, if he's under complement blockers, can have an optimal response in case of infection. So that's the reason why we're giving vaccination plus penicillin.
Right. Another question is crovalimab superior to revolizumab?
No, this is exactly the same drug. So data that has been presented are giving the same results. There is no randomly comparison that is ongoing. This is a Commodore 1 and Commodore 2 studies. I did not present them. But it's basically the question. It's revolizumab against crovalimab, randomized way, naive patients and patients who are already receiving complement blockers. If you go to trial.gov, you will find them easily. Commodore, COMMODORE. And we will have the answer with those studies, but I guess it will be non-inferior.
Another question is, are there any ongoing studies combining two proximal complement inhibitors?
Not yet. And they are independent companies, so I guess that nobody will do this study. And in a way, there is no reason to do it, because what you want to do is, as you want to as a combo, is possibly to use eculizumab plus proximal blockers to improve results. That I don't find anything smart in, because the patients don't want to receive two drugs. But that's the only one that might be interesting. For the both blocking at the same level with many drugs is not so interesting.
Thank you. Another question is related to your setup. When you have access to all these drugs, do you ever offer transplant to these patients?
Yes, we are offering transplant in case of a plastic anemia, because those PNH patients may evolve to a plastic anemia. So if they have a sibling, if they are young, we are doing transplant. We are offering transplant for patients who are transformed. I told you this is a risk for clonal evolution like MDS, AML, and then of course there is transformation. So we need a transplant. And of course, in case of clonal evolution, categorizing MDS like monosomy 7 or others, we are giving transplant. And we have a paper that has been published in Hematology, I think a couple of years ago, that was dealing with how to manage eculizumab in terms of transplantation. And we are giving the last dose of eculizumab at time of conditioning regimen, and then afterwards. The patient is 100% donor chimerism, so he doesn't need any more eculizumab.
The question is there data on haplotransplanting these patients?
Yes, there is some data coming from US. Rick Childs published 10 years ago. It was a BMT paper, I think, showing nice data on haplo with reduced intensity conditioning regimen. Few patients published yet. I guess that what we are doing is that when we need this kind of approach, the patients are more at the aplastic status. We're not transplanting hemolytic patients, low responders to eculizumab, because there is proximal blockers for this. So it's really when aplastic or clonal evolution. And so we are at the moment using the Baltimore approach. Blood advances, the first author is Amy Dizern, the last one is Brodsky. And it's Baltimore with ATG at the very beginning, day -9, 8, and 7, 2.5 milligram per kilo total dose. And so we are using this approach, which is working quite well for patients, plastics.
So another question is increasing PNH clone related to increasing risk of MDS or other clonal marrow diseases?
No, we don't have this enhance, and the data have been shown that it's not the clone size that is predicting the risk of clonal evolution. It's really the disease per itself, it's the aplastic anemia behind that is predicting the risk.
The question is back to anticoagulants. Are the newer anticoagulants direct acting used in PNH with thrombosis?
Yes, and the results are exactly the same. There is no better anticoagulation than another one. It can be warfarin or oral anticoagulation, low weight heparin, whatever you want. The risk is the same and the efficacy is the same.
Another question is, in atypical hemolytic uremic syndrome, where we have to use eculizumab urgently and we cannot wait for 2 weeks, then what do you do about the pneumococcal vaccination, etc.?
So of course, there is sometimes patients who have to be treated urgently. So we are giving them quinolone that are protecting them against meningitis and treating them up front. I mean, of course, yes, the HUS situation, but there is also the Budd-Chiari syndrome, the mesenteric thrombosis in PNH. We are treating patients up front quite quickly. So we're giving a large spectrum antibiotics, and most of the time it's quinolone, treating the patients, vaccinating at the same time, and then stopping 15 days after, and coming back to penicillin prophylaxis.
That was the last question from the audience. Anything here? So I think everybody is very happy with all their queries. Before I hand over to Dr. Manoj Kumar for the official vote of thanks on my behalf, thank you so much, Professor Latour, for being with us repeatedly and educating us. The quality of patients will definitely improve on this side of the world. Over to Dr. Manoj.
Thank you, sir. Thank you, Professor Dr. Regis Peffault. I think me and you share one thing in common, sir, that kids walk in, at least when you're finishing your lecture. So you were just wonderful, and we all in India are grateful to your talk, sir. Professor Dr. B Agarwal, sir, for pulling this blinder once again. Happy Teachers Day to you, sir. And our chief guest, Professor Prantar Chakrabarti, especially for putting hematology before everything. And two guest speakers, I already thanked Professor Regi and Dr. Shaji R Velayudhan from CMC Fellow, and expert discussions from 8 of them, Colonel Venkateshan, Dr. Pooja Prasad, Dr. Aditi Shah, Dr. Reema, Dr. Dibya Jyoti Prusti, Dr. Nakul, Dr. Mona, and Dr. Akshya. Thank you so very much for making it so lively. Sir, today, once again, we have 400 plus login, 400 plus doctors across 7 countries today: India, Nepal, Bangladesh, Singapore, Sri Lanka, Myanmar, and Malaysia. Thank you so very much for your presence, and all of you have a wonderful Sunday, and all the very best. Stay safe and stay healthy.
Thank you, sir. Thank you, Dr. Manoj.
Thank you very much. Take care and stay safe. Bye-bye.